Okay, so hi everyone. This is the lecture on body fluids and acid-base balance. So I'm just going to go through the core specification points that you'll need to know for the exam. And yeah, you can go into more detail, but these are a good baseline knowledge. So here's the Socrative login. So we'll start off with the basics about the volume and osmolarity. So we've basically got three main compartments. We've got our plasma and our interstitial fluid, which is part of our extracellular fluid volume. And we've also got our intracellular fluid volume. So our total volume is 42 litres. Our intracellular is 28. So that's kind of most of it. Then we've got our extracellular fluid, which is 13 litres. And that's divided into the plasma and the interstitial. And then we've got transcellular fluid, which is one you mustn't forget because that's our extra 1 litre of fluid. So these are just kind of numbers to learn, unfortunately. You'll have to keep coming back to them. But if you just kind of break it down, you go over it a few times, it should be pretty easy to remember. So another key point is our intake and output of water per day. So we take in around 3 litres from food and drink, and we lose around 2.5 to 3 through various mechanisms, breathing, sweating, in our feces, in our urine. So the key thing for lots of MCQs in our exams is about our electrolyte composition and our osmolarity. So osmolarity just means the concentration of a solution expressed as the total number of solute particles per litre. So that's a confusing way of saying just basically the concentration. So the higher the osmolarity, the higher the amount of electrolyte you're going to have in your given volume. So the numbers in particular to remember are going to be in this table. So we've got our sodium, which is mainly an extracellular ion. So it's mainly outside of our cells. Whereas with potassium, it's mainly inside our cells. So we've got 140 inside as opposed to sodium, which is outside. And I remember this as not very physiologically accurate, but the sodium is 2 letters. And it's 2 letters, so it means it can't get through this wall. It's a bit more bulky than the 1-lettered potassium. Just a nice way of remembering it if you get stuck. So calcium is also an extracellular ion mainly. So that fits that kind of rule as well. And the concentrations here are really important to remember because our extracellular concentration is much, much bigger than our intracellular one. So note the micromolar as opposed to the millimolar. So those are really important ones to note. As well, with chloride, we've got a higher extracellular concentration, which is 100 millimolars, and a variable intracellular concentration, but it is lower most of the time. Looking at the bicarbonate as well, we've got it on the outside mainly, whereas with our protons, it's mainly in the inside. So we've got a higher concentration of protons, which gives us a lower pH. And note here that it's nanomolars as opposed to millimolars for most of them. So yeah, again, these kind of fit the rule with the single letters getting through the cell, whereas the more bulky ones are kept on the outside. Not physiologically accurate at all, I'll stress again, but a good way of remembering it. So another key point is to know that the plasma osmolarity is 290 milliosmoles. That's a really important number to remember. And our plasma glucose, when we're fasting, so when we haven't had food for a while, is 4 to 6 millimolars. Right, so I'll skip the Socrative for now, and we'll move on to our maintenance of intracellular and extracellular pH. So this is really important for our body because we need to maintain a really tight range in our cells in order for our proteins to work. So I'll go into that in a bit more detail. So our pH is defined by this equation. So this looks a bit messy at the moment, but if you break it down, it just means pH is equal to the log of 10 of the hydrogen ion concentration. And that can be rearranged to be the hydrogen concentration is equal to 10 to the minus pH. So that means you can work out the pH or the hydrogen concentration from any numbers that you're given in an MCQ. So it's really important that you learn both of these. Our normal arterial hydrogen concentration is roughly 40 nanomolars, and that gives us a pH of 7.4. So it's a slightly alkaline pH. So as I said, this must be maintained in order for our proteins to function. And this is because our proteins are really sensitive to being ionized or non-ionized. So this just means ionized is the addition of a proton, non-ionized is the removal of that. And our proteins are very, very sensitive. The structures are determined by these very specific interactions between all of the molecules. And if that's disrupted, we get a change in active site structure, and that can impair their activity and therefore the functions. We really need to know about the Henderson-Hasselbalch equation here. And this is important for maintaining this range of 3.5 to 4.5 pH. So the equation here is pH equals pKa plus log 10 with A over HA concentrations. And we'll talk about what that means now. So our pKa is our acid dissociation constant. And you won't be expected to know these. You'll usually be given them in a question, so don't worry about that. So the HA is basically the acid that we're concerned about. And the A minus, is just the concentration of the conjugate base. So the conjugate base is basically the acid minus the proton in most cases. And we'll talk about when that isn't the case. So let's have a look at the arterial blood. What is the pH? What determines the pH of that? So the acid and the base that we're mainly going to be concerned about are CO2, which is an acidic gas. There's no H, which is a bit confusing, but as you know from your respiratory physiology, CO2 is an acidic gas, which really could disrupt our cellular function if we don't get rid of it in the lungs. The bicarbonate is our base. So that's our conjugate base as referred to our acid, CO2 acid. So here we've got our base, A minus, our HCO2 3 minus, which is 25 millimolar, as we talked about before. And then we have our acid, which is CO2, and that's concentration usually in arterial blood is 1.2 millimoles. So our pKa for this is roughly 6.1. Again, you'll probably be given that and not be expected to learn it, but you might want to check your individual university specification for that. So let's work this out. So we've got our pH here is equal to our pKa, 6.1, plus the log 10 of our concentration of our alkaline base, our base, which is 25, over the concentration of our acid, which will be 1.2. And that will give us a pH of 7.42, so that's within our range, our nice range up here. So that would be of a typical person at rest. Great, so how do we defend against changes to that, of the pH being in that range? So there are basically three methods. The first one is using buffers. The second one is using ventilation. And our last one is using the renal mechanisms. So we'll talk about each of these in turn. So the buffers include proteins, bases, and phosphate ions. Bicarbonate and phosphate ions. So our proteins can include things like hemoglobin, a really important one. And these basically take up or donate protons depending on the surrounding concentration of protons. And this helps to maintain a constant pH. So as you might remember from our discussions about hemoglobin, hemoglobin takes up protons and it kind of hides them away from the surrounding cellular milieu, and means that the pH, the protons that are bound to the hemoglobin no longer affect the surrounding pH. So another example would be HCO3, which also buffers protons in the lungs. So it takes them out and becomes carbonic acid So the ventilation. The rate of ventilation is proportional to the plasma CO2 with a constant respiration. So that basically just means if you're breathing really fast, hyperventilation, you're going to get rid of more CO2 and you're going to get hypocapnia, which just means less CO2. So then our last mechanism, the most important one, is our renal mechanisms. So let's talk a bit about that in more detail. So there are three methods which we will talk about. So here we've got the renal mechanisms which remove our non-volatile acids. So this is basically any acid that isn't CO2, any acids that can't get blown off by the lungs by hyperventilation. And these acids tend to be produced by the metabolism of sulfur containing amino acids or cationic amino acids. And we get acids such as sulfuric acid and hydrochloric acid being produced from these processes. And these acids really can't be blown off by the lungs. We can't get rid of them by ventilation. So there are three methods for this. The first one is reabsorption of filtered bicarbonate. So that's kind of adding base back into our blood. The second one is generation of new bicarbonate. When we can't reabsorb quite enough, we need to make a bit more. Our last method is secreting HCO3. So that would be when we have a bit too much bicarbonate in our blood and we need to get the acidity back up again. And we can do that by removing the base. So let's have a look at this. We've got the reabsorption of filtered HCO3. So our bicarbonate is freely filtered at the glomerulus and needs to be recovered. That is the main problem that we have here that needs to be resolved. So our H+ reacts with our bicarbonate here in the tubular fluid. And this is transformed into water and carbon dioxide by the action of carbonic anhydrase. So this is really accelerated here, this step, by this enzyme. So it's really important that this is found on this apical membrane of our tubular fluid. So then the CO2 diffuses freely across this barrier and reacts with our water again. So then this process reverses completely. We've got more carbonic anhydrase, forms more hydrogen ions and more bicarbonate ions. Our hydrogen ions are chucked back into our tubular fluid, and this can be by ATPases or by these apical NaH exchanges. So our bicarbonate is then returned to the plasma via other basolateral exchanges. So we've either got this NaHCO3 minus co-transporters or symporters, or we can use these chloride exchanges. And the aim of these is basically to ensure that we have an equal isoelectric effect. So we're not totally just losing all of our negative ions, we're actually maintaining our charge across the membrane by also transporting in some chloride ions, which are also negatively charged. And in the same way, we're transporting out some sodium, which is a positively charged ion, so we're maintaining our isoelectric balance. Okay, so second method, we're going to generate new bicarbonate in the type A intercalated cells. So this is where the CO2 and the H2O are taken up from the basolateral surface, so here, and they're converted to HCO3 and H by the carbonic anhydrase. So we get they diffuse across this membrane. The carbonic anhydrase converts them into our hydrogen ions and our bicarbonate ions. The hydrogen ions are then excreted into the apical surface, where they are bound to a buffer. So this is important because it means that they're kind of trapped on this side. Whereas with our bicarbonate ions, they're transported across the BLM into our plasma. And again, we've got this isoelectric exchange, we've got our chloride ions being converted across to make sure we get that equality of the negative charge movement. So yeah, as I said, our buffer is trapped in the urine by the buffers. And our buffers can either be phosphate or ammonia. So the phosphate is dietary in origin, so it's quite variable in its availability. We're never really sure how much we're going to have at any one time. Whereas with ammonia, it's actively synthesized in these tubular cells by a process called Ammonia Genesis. And that can be altered to reflect this acid-base balance. It can be upregulated if we need more, or downregulated if we don't need as much. So ammonia genesis just means the deamination of glutamine. So these are the steps. We've got glutamine converted to glutamic acid, converted to alpha ketoglutarate. And at each step, we're forming an NH3 and an HCO3 minus. So we're forming our base in this process. Okay. And our last mechanism is the distal tubular secretion of bicarbonate. So this happens in type B intercalated cells, as opposed to the previous method, which was type A. These cells secrete bicarbonate during alkalosis. So I remember this as the B is for when it's basic, when there's a lot of base around. They basically have the same configuration as the type A cells, but reversed. So we've just got swapping of the transporters from the apical to the basolateral membrane. Normally, there are far more type A cells, but when it's alkalosis, as we said, when there's more base around, they're significantly upregulated. So the steps in this process are the carbonic anhydrase forms our H plus and our HCO3 minus again. Really accelerates that step. And then the HCO3 minus, rather than going across the basolateral membrane, goes across the apical membrane. And that's an exchange again for our chloride. And then the H plus, they are reabsorbed into our plasma via these ATPases. So I don't know whether we can do this Socrative. I think I'll skip it for now, because I'm not sure who's here, but we'll carry on. So yeah, I just thought I'd mention a little bit about assays. This has been a very short lecture. There's not much to cover in this topic, but hopefully this was a really good overview. For those of you at Oxford, this will be useful for your specific essays. For those of you not, it might be useful in general. So my top tip would just be make sure you make a plan. Don't just start writing. Use 5 to 10 minutes just to really make sure you know where you're going, what you're doing with your essay. You also need at least 5 points which fully answer the questions. So this is the really important point. You need to fully answer the question. If you haven't covered the basics, you're not going to get the marks. So you need to make sure that you've got the basics down before you start to expand. So if you do choose to expand, you can add experiments or clinical examples which back up your points. And this again is the really key part. You need to make sure that they back up your points. You're not just throwing them in at random for the sake of it. They need to be really relevant and really structured. And another tip, just time yourself really, really strictly. Ensure that you don't spend half an hour on your introduction and then have no time to complete the rest of your essay. This applies for MCQs as well. And yeah, again, just try and cover all of the points on your main syllabus, because if you haven't done that, there's a limit to how much credit the examiners can give you. Great, and just keep calm. Just keep going. If you've forgotten everything about medicine, or it feels like that, you haven't. There's something in there that's going to be useful to you. Just keep going no matter what. Yes, so this is a Socrative quiz just to accompany the lecture. I'll just go through these. So approximately how many litres of plasma are there in the body? So just have a go answering that. I'm going to put in fake answers just to go along with you guys. So let's have a look at the results now. So yeah, it's 3 litres If we have a look at the explanation, it's basically there's 13 litres of extracellular fluid, 3 litres of which is plasma. So nice easy one to start. Let's have a look at this question. So how many litres of intracellular fluid are there in the body? So have a guess. Can I have a guess? So it's 28 litres. It's pretty self-explanatory, just going back to that slide that we looked at with the different body compartments. Right, next question. What is the approximate daily intake and loss of water per day? So the answer for this one, you can pause the video if you like, but the answer for this one is 3 litres, intake and daily loss 2.5 to 3. So what is the value of plasma osmolality? You can remember this. So the answer is 290 milliosmoles. So there we go. Next question. What is fasting plasma glucose? So this question, true or false, 4 to 7 millimolars. Now let's have a look. It's false. So looking at our explanation, it's 4 to 6 millimolars fasting. Let's keep going. So what are the correct values for extracellular sodium, potassium and calcium respectively? So just have a guess. Think about our extracellular sodium, so it's Na, and our potassium, which is intracellular. Let's have a look. So yeah, we've got our 144, 2.4. So that's just a matter of learning those figures. Okay, let's keep going. What are the intracellular values of bicarbonate ions and protons? So thinking about, again, we've got our bigger ion and our smaller ion. And our answer would be 15 millimolars and 75 nanomolars. So that's just, again, a bit of rote learning. You either know it or you don't. So yeah, just a good one to have on your wall, maybe to stare at when you're a bit bored. Let's move on. So potassium is primarily an extracellular ion. True or false? Give me a few seconds. So it's false. Potassium is primarily an intracellular ion. So we've got 140 intracellularly, remember, because it slips through the wall, and 4 extracellularly. Okay, moving on. So as ventilation increases and more carbon dioxide is removed from the blood, what happens? Will CO2 increase? Will the hydrogen ion concentration of the blood decrease? Will the blood pH decrease? Will the hydrogen concentration of the blood increase? What do you think? So a few more seconds to think about that, and let's have a look. So the hydrogen ion of the blood will decrease because, as we mentioned, our carbon dioxide is an acidic gas, and so it will increase our hydrogen concentration when present, but if it's decreasing, it will decrease our hydrogen ion concentration. Okay, our next question. So a major hydrogen ion buffer in the blood is what? What do we think? Albumin, carbonic anhydrase, hemoglobin, creatinine, or urea? A quick guess. So hemoglobin is our main one. Let's keep going. So efflux of bicarbonate ions at constant intracellular and extracellular CO2 will do what? Will it increase intracellular pH, decrease intracellular pH or have no effect? What do we think? Getting rid of bicarbonate ions. The answer is it will decrease the intracellular pH because we're getting rid of our base, which means we'll be lowering our pH, making it more acidic. Next question. If the pH of the blood becomes 7.49 due to ingested substances, all of the following would happen to compensate, except what? So the blood is becoming more alkaline. What would happen? Would the ventilation rate decrease? Would the kidney increase the secretion of bicarbonate ions? Would the tubule cells produce more ammonia from glutamate? And would the partial pressure of carbon dioxide in the blood start to rise? Have a quick think. So it's going to become more alkaline. What are we going to do to compensate? What isn't going to happen out of these? So the answer here is, oh, computer just crashed. The tubule cells producing more ammonia from glutamate. And this is because when we produce ammonia, we're producing bicarbonate ions. So that's going to make our blood more alkaline. So if our blood is becoming more alkaline anyway, we're going to want to reduce that effect. So the ammonia genesis is going to decrease. Right, next question. In response to metabolic acidosis, the body will what? Excrete more bicarbonate, increase the ventilation rate, decrease the ventilation rate, or decrease ammonia genesis? What do we think? So we've got acidic blood. How are we going to make it less acidic? The answer here is increasing the ventilation rate. It's the only process here that's going to make our blood more acidic because, less acidic, because we're getting rid of our carbon dioxide. Here we're getting rid of our base, which is just not what we want to do. Here we're retaining more CO2, which is also not what we want to do. And here we're making more, we're making less base, because as we said before in the last question, ammonia genesis generates bicarbonate. Hope that makes sense. Next question. So generation of new bicarbonate typically occurs in type A intercalated cells. Is this true or false? So type A, when were they most present as opposed to type B cells? Think about that. And let's see what the answers are. So this is true. They generate new bicarbonate because they tend to be present when there's less base around, as opposed to type B cells where they're more present when there's more base. Okay, so what's the approximate concentration of hydrogen in the blood? So just think about our equations that we know for this question and try and make an approximate answer for that. Let's have a look. So it's 10 to the minus 7 moles because we're using our pH of approximately 7.35 for that. Next question. Inhibition of carbonic anhydrase will do what to sodium reabsorption? So this is a bit of a tricky one. So think back to our cells that we looked at. And just think about which transporters affected our sodium reabsorption. So a few more seconds and then the answer is decreases. So this is basically because our carbonic anhydrase makes bicarbonate, and that bicarbonate is reabsorbed across the basolateral membrane, sometimes in association with sodium. So if we're making less bicarbonate, we're going to reabsorb less sodium. So just go and stare back at the slides if you're confused about that. So here are just some essay questions. I won't dwell too much on these, but if the essay was called Compare and Contrast the body's response to acidosis and alkalosis, just have a little think about what you'd want to include in that essay. So some ideas here. You'd probably want to define what normal blood pH is and why it's important that we maintain it, which I hope we've covered in a lot of detail in this talk. And then we're going to want to talk about the 3 lines of defence. So we've got our buffers, the ventilation, and also our renal mechanisms, which will require a bit more explanation. And really think about the wording of the question. So you're comparing and you're contrasting. So you need to think of specific points that are going to allow you to do that and to show that you can compare and contrast, because that is a skill that it sounds easy, but it's not necessarily so. And yeah, just adding some extra detail where you think is appropriate and where it's useful. And our last question, what is the renal contribution to plasma pH regulation? So that here is just dwelling more on the last of those mechanisms that we talked about before. So we're going to talk about the reabsorption of filtered bicarbonate, the new bicarbonate, and the distal secretion. And you go into detail, maybe draw some diagrams of those processes, just showing them that you really understand what's going on. And remember to cover all of those basics before you go on to add the extra detail. I'll say that again because it's very easy to skip out the basics, and that means that you drop all of the marks, even though you've shown that you know a lot more about other stuff. So there we go, that's the end of the talk.